Good afternoon, everyone, and welcome to a new seminar of the EBD Seminar series. Before I introduce our speaker today, just a couple of reminders. You can write your questions for Christian in the chat. And also, if you need a certificate of attendance, please write your full name in the chat and send an email to seminarios@ebd.csIC.es. Infectious diseases are one of the major challenges in our society, as we have all suffered this year. And monitoring and understanding wildlife diseases is key. Our speaker today, Christian Gortázar, is a world expert in disease ecology. He is professor in the animal health area at Universidad de Castilla-La Mancha. He is PI of the Health and Biotechnology Group at the Instituto de Investigación de Recursos Cinegéticos, and he is part of several expert committees, including the Food and Agriculture Organization of the United Nations. So without further ado, Christian, you can start whenever you are ready.
Thank you very much for the invitation. It's a pleasure really to be here and to speak a bit about TADs, transboundary animal diseases. So what is this? Essentially, these are diseases that have a strong impact on the livelihood of many people, particularly in developing countries. But in addition, these are diseases that may emerge and that may jump over to other geographical regions. And in fact, many of the TADs are currently expanding their geographical range. And there is a risk of them entering Europe. And in fact, some of them have already entered several times. How do these pathogens get introduced into Europe? Well, essentially, it can be through commercial movements, and this has enormous implications on commerce, on economy. But they can even be introduced over the wind, or through fomites, or through human beings carrying contamination, and obviously also through wildlife, for instance, migratory birds. And once they enter, some of them can be of catastrophic consequences. Just think about the foot and mouth disease outbreak that they had in the UK in 2001. This had enormous consequences on the economy and even consequences on other diseases. But what is the role of wildlife in all this? Regarding transboundary diseases, wildlife may be susceptible, but then we don't know if it's a reservoir, if it's an indicator, or if it's even a victim. So the idea of this presentation is first to introduce a number of these transboundary diseases that we call fast diseases, then review shortly 6 of those fast diseases and the role of wildlife, and with this in mind, think about wildlife and disease surveillance and the role of wildlife regarding disease control. So that's what we are going to do. Let's start with introducing the fast diseases. This fast comes from foot and mouth disease and similar transboundary animal diseases. So you see, it's a very complicated acronym. The idea is that there is a strategy, an international strategy, to try to stop the emergence of these kind of diseases. And this is for several reasons, but mainly because they have a strong impact on economy. There are different categories of fast diseases, and we are only focusing on the first 2, on the more relevant diseases, and these are 6 in total. And our question for today is, what is the role of wildlife? Is there no rule at all, or do they have what kind of rule, and how does this interfere with disease control and affect wildlife management in general? So let's go disease by disease, starting with possibly the most important one, foot and mouth disease, or FMD. As you probably know, because foot and mouth is the most well-known of all these problems, this is a viral disease that is transmitted both directly and indirectly, and that can even be carried by animal products. This means that, of course, if you have a foot and mouth outbreak in your country, your livestock production is essentially stopped and your exports are essentially completely stopped. So the hit to the economy is tremendous. And at least in some circumstances, wildlife are important because they constitute reservoirs for the virus, where the virus can survive even in the absence of livestock. And unfortunately, foot and mouth disease is continuously expanding from different areas where it is endemic to, so to say, newly colonized areas. And some of these areas are pretty close to Europe. Nowadays, this includes Turkey, this includes Morocco, for instance. And wildlife can be implicated, not only African buffalo, also other species, like, for instance, impalas, can not only carry the virus, but also be victims of the disease, as you can see in these photographs here. However, the most important or the best known wildlife reservoir for foot and mouth disease is the African buffalo. And we will come back to this at the end of the presentation because there is some very, very interesting research going on in this regard. And it is also a means of introducing the collateral effects of these kind of diseases on conservation. For instance, many of you know that in the southern tip of Africa, there are large fences, for instance, separating Botswana from South Africa, Namibia from Botswana, or the Kruger National Park in South Africa from the remaining part of the country. These kind of large-scale fences are in place in order to avoid the movement of buffalo and other foot and mouth disease reservoirs. So obviously, they will have a consequence on animal corridors, on habitat connectivity, and therefore on conservation. From a European perspective, European wildlife can be infected with foot and mouth disease virus. In fact, there have been lots of experimental infections in deer species showing that they are susceptible, and in fact, they have been found infected in several places. And wild boar are essentially pigs, so they are also susceptible, and they can act as breech hosts. We will see it later. However, when the big last outbreak of FMD happened in the UK in 2001, causing a huge loss and among other things, 7 million culled animals for disease control, the fact is that no wildlife control, no actions on wildlife were taken. And despite taking no action on wildlife, the foot and mouth outbreak was controlled. So in principle, a minor rule for wildlife, if any. And more recently, a bit more recently, in 2011, there was a second outbreak in Europe, in this case in Bulgaria. And this was a border region with Turkey. Here, the first case was discovered in a wild boar, and then there were cases discovered in domestic animals, cattle, goat, sheep, pig. And again, many animals were culled. There was no specific action on wildlife. And despite having sporadic cases in wild boar and in roe deer, again, the problem was controlled. So after these experiences in the UK and in Bulgaria, the view was or is that foot and mouth disease infection in wildlife is a short-living event. However, the interesting point is that at least in European settings, it really seems that the rule of wildlife as breech hosts can be relevant. In the case of Bulgaria, apparently these first cases in wild boar may be due to cross-border movements, could have been due to cross-border movements, and very recently, this month, there have been outbreaks in Israel at the Lebanon border, and again, wild boar has been identified as a possible breech host in that case. Now let's go to another completely different disease, but somehow related, because again, the main actor are ruminants. This one has a French name, Pest de petite gumina, PPR, and it is a morbillivirus like carnine distemper or like so many others. So they are very directly transmitted. They cause huge mortality, And there is a strategy, a global strategy for eradicating this virus. This virus does affect wildlife. This is well known. There are many artiodactyls, including ovines, caprines, bovines, gazelles, and so on, that have been found infected. And even white-tailed deer, so a cervid, has also been experimentally infected, further expanding the range of hosts. So it can affect many wild ungulates. And in fact, if you map it, you will see that there are implications of wildlife in many different parts of the world. The interesting thing regarding PPR is there is a clear effect on conservation. You all probably know about the fate of the saiga antelopes in many parts of Asia, which has to do with outbreaks of PPR. Another disease, the third one, lumpy skin disease. This is a pox virus, a Capri pox virus, that, as opposed to the other two, is also transmitted by vectors. It causes a relatively mild skin disease, but still it is a problem. And this one has eventually entered Europe. Here you can see the distribution of cases in a number of years, and this situation was essentially controlled through a massive vaccination of cattle, which is the main host and the main victim of this pox virus. So, in principle, since vaccination controlled everything, and since, well, there are not so many studies showing a relevance of wildlife, it is considered that wildlife does not play a meaningful role in the epidemiology of lumpy skin disease virus. However, if we go far back into the literature, in this year 70 of the past century, there were some experimental infections in giraffe and in impala, showing and suggesting that there may be a wild cycle of this virus, at least in some specific areas, in this case, again, the Kruger Park in South Africa. So who knows? Another very related virus or group of viruses is sheep and goat pox virus. Again, a pox virus like lumpy skin disease. And again, you will find it in domestic small ruminants, but you will find it also in wildlife, sometimes with a high mortality in the domestic hosts, particularly if they are not native breeds. Sheep and goat pox is very widely distributed. You can see all Northern Africa, Asia, and so on. So Europe is essentially surrounded by positive areas. And there is very little information on the role of wildlife or the consequences for wildlife, but there is at least one event where a familiar of the tar, the serow in India, was severely affected by a goat pox virus. So there are some risks for wildlife. However, probably they remain essentially unknown. Yet another one, Rift Valley fever, is very important because this is not only a vector-borne disease, but in addition, it is a zoonotic disease, meaning it can be also transmitted and clinically affect human beings. So the epidemiology is quite interesting, as you can see from this figure. There are epidemic events and there are inter-epidemic periods, like it happens for many other of these TIDs. And while during the epidemic events, there have been a lot of studies in the inter-epidemic periods, the information is a bit more limited. And it may be that wild animals, particularly ruminants, but not only, play a significant role in the maintenance of the infection during the inter-epidemic periods. Again, this is a geographically expanding virus where it is known that some wild animals are pretty relevant in infection maintenance. And in this case, it may even be that some non-artiodactyl mammals play a role. Maybe Chiroptera, maybe Rodentia. How is it spreading? Well, one route of spread is just the movement of live domestic animals. So these are relatively short-range movements happening everywhere and bringing the infection to many places. Currently, there have been relatively recent reports from Tunisia, and there were reports also last year in Libya, and of course in Egypt and so on. So it's not so far away. And again, wildlife, in this case wild-tailed deer again, have been experimentally infected, and it has been shown that in fact wild deer are susceptible, but not just susceptible, they also can transmit the virus in principle through direct contact to other individuals. And the last one of this list is bovine ephemeral fever. Bovine ephemeral fever is a rhabdovirus. The rhabdovirus, you know, is rabies. So this one is not as bad. It just causes a drop in production, particularly in dairy cattle, but it is pretty extended over Africa, Southern Asia, and Australia. And obviously, there is also concern in Europe, particularly because it is known that several wildlife species, like this waterbuck, for instance, can act as maintenance hosts for this virus. And the virus is also able to infect cervids, including our European red deer, as it has been shown to infect several Asian and Asian cervids, and also red deer or equivalents in Australia. So, to summarize this story about the different diseases, even if you don't know the names, don't care about that. But the first conclusion, I hope, from this overview is that we can discard the first option. There is a rule for wildlife, okay? Depending on the disease and depending on the setting, the rule may be just as an indicator, just as something that we should sample in order to know if this is telling us something about the presence, absence of the disease, or in some cases, wildlife may be victims, may be bridge hosts, or may even be maintenance hosts. So, knowing this, what is the role of wildlife in disease surveillance? Well, first thing is, wildlife is very useful for early detection. Remember that in the case of the foot and mouth disease outbreak in Bulgaria, wild boar was the first indicator that something was going on, and later foot and mouth was discovered in the domestic animals. But the first indication was a wild boar that was shot by hunters, but it was shot because it was lame. It had some clinical signs that were not usual. So early detection is one of the very, very interesting features of wildlife disease surveillance. Second point is wildlife can be sentinels. If we are vaccinating all the livestock will have antibodies. And unless we have a specific diagnostic tool that distinguishes between vaccine antibodies and disease antibodies, we no longer know if an animal has been infected or just vaccinated. This is the case for PPR, where vaccines are not DIVA, meaning vaccines will produce the same antibodies that you find under field conditions. Okay, so having wildlife populations in the same area and sampling them will tell you if there is real circulation of the virus after the massive vaccination campaign. And finally, it is just that in the 21st century, it is impossible to consider a disease surveillance scheme that is excluding wildlife. And it needs to be integrated, meaning that it should focus not only on livestock and wildlife, but also on the two aspects, health and numbers. We need the health as the numerator, and we need the numbers as the denominator for any epidemiological analysis. and think that risks for wildlife go far beyond the direct effects of the mortality caused by a disease, say, for instance, PPR and saiga antelopes. Think that pathogens change, wildlife populations change, and there may be just sporadic jumps, and there may be effects that we don't foresee. For instance, in the hares, now there is a myxoma virus circulating and causing some mortality that in fact includes a small part of another virus, which happens to be probably a pox virus of a ungulate. So you see, pathogens tend to evolve as hosts do, and unfortunately, probably at a faster, much faster speed, with some consequences eventually for conservation, for management, and for disease control. Then, considering all this, what is the role of wildlife in the control of fast diseases? Well, on one side, wildlife themselves, they can favor the abundance and survival of vectors. For instance, it is known that culicoides midge populations in Spain are in part, their abundance is in part mediated by the abundance of wild animals. In the case of Aedes mosquitoes in some parts of Africa, there is also evidence suggesting that the abundance of wildlife hosts is driving the abundance of the vector. And in the case of ticks, it is even more evident because, at least in European countries, tick control in livestock is something that is quite usual. So if you are controlling ticks in the livestock through antiparasitics or whatsoever, but you are not controlling them in wildlife, obviously, then probably wildlife is having a much higher, a much more relevant role in the maintenance of the tick populations. Then, as I said when starting the presentation, wildlife can transport pathogens or vectors. For instance, migratory birds do, and you know this pretty well in your team in Doñana. And this is often something we tend to forget about. Wildlife can act as bridge hosts for livestock pathogens. A bridge host is something that is not maintaining the disease necessarily, not for a long time, but maybe a few infected individuals or just one infected individual can make the bridge between, say, the wild environment and a farm, or between one geographical region and another, as it happens right now with the case of wild boar between Lebanon and Israel, for instance, and foot and mouth disease. And eventually, in some cases, wild maintenance hosts will obviously interfere with disease control. This is the case of the endemic areas of foot and mouth in Africa as the best example, but probably it is not the only case. And the problem is that we just don't know exactly what is the role of wildlife in several other settings and diseases. The important point here is that the role of wildlife will not be always the same. It depends on densities, it depends on the species composition, it depends on several factors. And obviously it is dynamic, meaning that if populations change, their relevance will change in turn. This we know it pretty well from blue tongue. Blue tongue is a vector-borne orbivirus that is transmitted by culicoides, and that is essentially infecting African wild gazelles and wild ruminants. From there, in historical times, it spilled over to domestic ruminants, mainly sheep, and it has now become a problem that is almost global. Well, blue tongue can or cannot be a problem regarding wildlife, depending on the epidemiological situation, depending on densities, depending on climate, depending on a number of factors. So the rule of a red deer in blue tongue maintenance is not the same in Spain as in Belgium, for instance. And even more complex, this is a fantastic pseudo experiment that was carried out in the Kruger Park again with buffalo. In this case, people were studying buffalo and tuberculosis, and it happened that there was an outbreak of Rift Valley fever, so one of those epidemic events. And they realized that the bovine TB positive buffalo were more affected by Rift Valley than the bovine TB negative ones. When they modeled this situation, and this is what you have to the right side of the slide, the small dotted line that lies below indicates what would happen with the Rift Valley fever outbreak in case there was no TB, and the dashed line on top is showing what happens with Rift Valley fever in case of hitting a population, a wildlife population in this case, that is severely affected by tuberculosis. As you can see, the effect of the viral disease of Rift Valley is much stronger when the bacterial disease, the chronic disease, tuberculosis, is predisposing the hosts for a stronger impact of the disease in terms of infection, in terms probably also of producing new viruses and contributing to transmission. And now make a parallelism with your main study area, Doñana National Park. In the Kruger Park, about 50% of the buffalo are TB infected naturally. But unfortunately, this is also the case of red deer or wild boar in Doñana National Park. So imagine there is an outbreak of Rift Valley fever, which is not so far away, which is not so impossible to conceive. So probably the impact in a TB endemic population like the one in Doñana could be predicted to be stronger than the impact in a population where TB was not circulating at the same level. And now we are starting to get to the end of the presentation. This is a very simple and stupid graph showing more or less the scientific production increase from 2000 to 2020 in the last 20 years. Okay, so the mean increase of all scientific fields is about by 4 in these 20 years. So the research on these 6 diseases that I presented you, that I introduced in this presentation, has been a bit faster, has been a bit more. There was a growth by 6 in the same time. And if we focus specifically on research on fast diseases dealing with wildlife, then the increase is spectacular. It's a 22 times, okay? But the reality is that still, if we take all the literature that is available, the part that is dealing with wildlife, that is considering wildlife, is just 10% of the whole information that is available. And that's clearly insufficient, as probably could be evident from some of the slides I showed you. And in fact, even the more recent report on the situation of fast diseases in the European surroundings does not mention the word wildlife at any point. So there's something to do about it. And final section. What do we need to do? Get ready. We are aware of some fast diseases, or fast related, if you want, that are very likely to emerge sooner or later. One of them is African swine fever. And for this, the veterinary services, the researchers, the farmers, everybody, the hunters, everybody is trying to get prepared, to get trained, and to at least be ready for whatever might happen at some point. And then we need to expect the unexpected. We need to consider that things might change from one year to the other. And this is exactly what has happened with the COVID-19 crisis. We predicted that there would be some effect on animal health. And in fact, this is already happening. The global efforts to eradicate PPR virus, essentially through massive vaccination, is already struggling, finding some trouble because of COVID-19. So yeah, who would expect this 2 years ago? But even more, the virus causing COVID-19, SARS coronavirus 2, happens also to be able to infect deer. This has just been shown by our colleagues from the USDA, by Mitch Palmer and his team. They inoculated the virus into white-tailed deer. White-tailed deer developed signs, some died, some survived. The interesting point is not this. The interesting point is that they showed in the laboratory that there was transmission between the infected animals and some in-contact animals. So this, if it holds also under field conditions, which we obviously still don't know, would allow the setting up of a wildlife reservoir for COVID-19. Okay, so don't be afraid, and just try to remember these few simple messages. First thing, there is a lot of global change and there is a lot of pathogen spread all the time, everywhere. So be prepared. Regarding wildlife, clearly they have a rule. They can play different rules. The rules will change from one place to the other. But the rule is there. It is just that we don't have enough knowledge to set it properly. And wildlife diseases and in general, multi-host diseases and multi-pathogen settings are immensely complex, and we need very, very, very much research in order to try to understand the drivers of disease emergence and to understand what are the circumstances under which, by making integrated approaches, we could be able to control certain diseases, never all, just certain very relevant diseases, through interventions, not only on domestic animals, but also on wildlife, and eventually, of course, also on human beings. Thank you very much.
Thank you very much, Christian. It's been a great talk. So we are going to go ahead and make some, I'm going to tell you one of the questions that people has. Okay, so here we have a question. Well, say thank you for the talk. It says, it seems that dispersal is fundamental for the persistence of a species, including disease-causing transmitting ones. Are there initiatives to deal with these transmissions without blocking wildlife dispersal?
Yeah, it's a problem. In some species, yes. I'm not sure if there are initiatives for everything, but clearly in some cases, there is use of reproductive technologies or of other means, or there is transport of individuals after checking and rechecking they are not infected, these kind of things. So yeah, it is of concern, and it is addressed at least for, so to say, the keystone or the more evident species, but probably the effects will go beyond those more visible ones. Mm-hmm.
So, okay, we have another question. So you commented for the LSD that there are it's also transmitted by vectors. Do we know which kind of vectors there are, like culicoides, Aedes, or ticks, or?
It is not completely clear because it seems that there is a broad range of mechanical vectors that can be implicated. The only one that is pretty clear, or seems to be more clear, is Stomoxys calcitrans, which is the stable fly.
Okay.
The blood-sucking fly.
Mm-hmm. Okay, let's see if we have more questions. Okay, so we have another question regarding surveillance. You were talking about how it is important to do surveillance in wildlife, but I mean, you are clearly very involved in research. But I don't know if there is established surveillance aside from researchers. You know what I mean? In wildlife, not in domestic animals. Yeah.
It's a growing field, and depending on the country, essentially, or the region, there is more effort or less effort. in most of the developed countries, there is some degree of wildlife disease surveillance. But as I said, we need a surveillance that is integrated, that integrates health surveillance with population monitoring. And this is something that is just starting. We are preparing with one of the students here at IREC a review on this aspect, because I think there are certain unknowns that should be addressed here.
Yeah, I mean, our experience with West Nile virus is the same. You need a better integration of the surveillance. So Agus is asking, what would be your advice to deal with the recent outbreaks of Nile fever?
Sorry, could you repeat?
What would be your advice to deal with the recent outbreaks of West Nile fever?
Why? Very difficult question. I would say, essentially live with it. I mean, there is no way in certain diseases, there is just no way to control them. So you need to adapt to them. So this means, West Nile does affect horses in addition to humans. Obviously, in the case of the horses, people are already vaccinating. So that's one obvious thing. And in the case of human beings, there is self-protection, awareness in the medical services, so that interventions are targeted as soon as possible, so that nobody with characteristic signs ends up waiting too long for a diagnosis, these kind of things. But otherwise, there is not so much you can do, unless you want to interfere completely with the environment, which is not an obvious step.
So yes, survey is one of the examples where surveillance is key to know where. Absolutely.
And you have very interesting tools. I mean, you even have testing the blood donors and these kind of things. So there are interesting tools for a real integrated surveillance, where you can have benefit from information that comes from the human side, from the animal side, from the wildlife side, and obviously from the vector side.
Yeah, so that's key. So Pablo Pastor is asking, what would be the approaches to the conservation of the wildlife regarding all these diseases? So
again, it depends on the disease. Many of these diseases don't harm wildlife too much. It is just when, generally, when there is a new entry of a very high mortality infection like PPR into a naive population happens, and only when this naive population has a very strong intraspecies contact rate, that you can have these spectacular outbreaks like you had in the PPR case in the Saiga antelope. So in general, I would not expect a big impact. I would expect a bigger impact from the control measures, like, for instance, fencing or these kind of things. So the indirect impacts are probably more relevant. In this case, at least in the diseases that I reviewed, I think the rule of wildlife is more interesting because of their epidemiological rule and because of the possibility of using them as indicators than because of the disease risk for the wildlife itself.
Great. So Carles Vila asks you, is it possible to manage or control the spread of the viruses in wild ungulate populations? I guess that it's impossible if the populations are not heavily managed. That's what he says.
And even if they are managed, it's very difficult, because you see some of them are vector-borne, some of them have multiple means of transmission. So it's really difficult. But the point is not so much should we or should we not. The point is, when we want to control a certain disease, we need to know what role is wildlife playing. And in some cases, the answer may be like you would have, for instance, in South Africa, to give the example. Okay, we know wildlife is maintaining foot and mouth, and in the areas where we do have wildlife, just don't think about eradicating foot and mouth, because it's just impossible. You cannot vaccinate all the wildlife unless you have an oral vaccine, which is not the case. But you have to consider this, and you have to probably regionalize your management of your cattle or of your livestock in order to consider free zones and infected zones. So essentially, There are different approaches that may range from direct intervention, including vaccination, through separating wildlife from livestock, over to doing nothing, depending on the disease, depending on the situation. But the important thing is not to play the ostrich, not to hide, and to know what's going on.
Yeah, that's really important, because I mean there are diseases that are really problematic in that regard. I'm thinking about the badgers in the UK, where everybody seems to fight between cooling or vaccination, or there seems to be this continuous fight of what should be the best approach. So I think what you're proposing, it's really like a better approach. I have a question regarding the myxomatosis in the hair. So you said that myxomatosis, that new strain, virus that is affecting the hares, do you know it's able to go back to the rabbit and infect the rabbits and then cause new disease in rabbits that were supposed to be already resistant to some extent?
I know it is being found in rabbits. I don't know if it has a strong effect on the rabbit population. I don't think so. At least it does not seem to be a very spectacular one. Anyway, I even doubt that the new myxoma virus has a very, very strong effect in hares. There was a strong effect, or there was a more evident mortality in the first year, maybe the second year of the outbreak. Right now, it seems that this was a wave that crossed the population, that caused some mortality, but that does not look like being the big cause for the hare decline. I think hare declines are much more linked to habitat change and to other factors than to this new virus. But that's a personal opinion.
Okay, well, we'll see what the research says. So I don't know if we have more questions here. I'm going to ahead and ask you what Agus was asking you before, regarding like what has been like your path in research and how did you get to where you are right now?
Well, essentially, I was very lucky. But the interesting thing is that I did not have a grant for doing my PhD, and I did not have a postdoc later somewhere, and so on. No, when I finished, well, actually, I started my PhD before finishing my studies. This is already a bit bizarre, but well, things happen, and the professor offered me the possibility of working with foxes, fox ecology, and fox diseases, and I was still in third course of 5. So I started with this, ended the career, well, more or less as I could, while doing some radio tracking with foxes and lots of field work. So, so I had no competitive marks for getting a doctoral grant. So when I finished the career, I set up a small consulting with some colleagues working on wildlife diseases and wildlife management. And remained in this consulting while doing my PhD, like, by the way. And 2 years after finishing my PhD, it happened that the IREC Institute was set up, and they were looking for a veterinarian with some background in ecology and so on. So it was just fitting my profile. So this is how I entered science more formally. So you see, it's not the usual way of doing it.
Great. Now it's good to hear that there are alternative paths to finding, and I think our PhD students and postdoc will find that really helpful, your advice. So thank you very much. Wait, there are a couple more questions while we were talking.
Okay.
So the example of FMD was interesting, compartmentalizing areas in Africa. Do you think this is something we may need to do with wild boar and ASF in Europe, or would it be possible to do it?
Wow, that's a very, very politically heavy question, because you can imagine just in Spain, there are over 300,000 employments depending on the pig industry. Okay, so imagine this at the European scale. The hit of African swine fever is something that is just unbelievable. So this said, the idea is that in general, the spirit of the new European animal health law is that whatever is there in wildlife does not necessarily mean it is officially there in domestic animals. So we are already kind of compartmentalized, creating these compartments between wildlife and domestic animals. Also, it depends what kind of entry you have of the disease, because the outbreaks in the Czech Republic and more recently in Belgium were completely eradicated after some interventions, which include essentially fencing, movement controls, some culling, and so on and so on. So it is possible eventually to control African swine fever under certain circumstances. In endemic areas, or more or less endemic areas, where you have the disease running for years over a large area, there it becomes more complex. But whether we should go for some kind of geographical differentiation or so, I think it is too early to say it, even after 6 or 7 years of fighting with the problem.
Mm-hmm. So we have one more question, which is actually related with what you just talked about. Monsevila says first, thank you for the talk. And she was asking, how do you explain the control of FMD and other outbreaks without major management actions?
You mean major management actions of wildlife? Well, every disease is different. That's the main point is, like every host is different, you cannot think of the behavior of wild boar in the same way as you think of the behavior of red deer, for instance. And in the viruses and bacteria and so on, it's the same thing. You cannot compare foot and mouth with African swine fever or with, I don't know, Rift Valley fever, because they just behave different. In the case of foot and mouth, probably one thing you need is very high density of the host populations for the virus to circulate in an efficient way. So it seems that if you really eliminate from the areas you are wanting to eradicate the disease, all the dense host populations, in this case in Europe, mainly the livestock, that then it seems that wildlife at a certain density is not able to maintain the circulation. It is just able to maybe carry the virus from one place to the other, as I explained with the wild boar in Bulgaria, for instance. Well, this is how it looks like for the moment, but we don't know what would have happened in the UK, for instance, if in addition to their deer populations, they would have had at that point already dense wild boar populations in some places. We also don't know what would happen if foot and mouth enters Spain with the very high wild boar densities we have in some areas. For instance, close to the city of Barcelona, the estimation right now is about 20 wild boar per square kilometer, and they are not uniformly spread. They are probably concentrated in the more food-rich areas and so on. So who knows? I mean, we can learn from what has happened, but we need to be prepared for a different setting and a different host composition, a host community having a different response.
Yes, because aggregation tends to increase, well, for some diseases, obviously, but aggregation increases like transmission of diseases. Well, it happens in distemper in raccoons.
Exactly.
And raccoons, like when you have them aggregated in urbanized areas, like there is like an increase in rabies, there is an increase in canine distemper and so on. So aggregation and a different pattern, like with the boars and everything, it's really like it's a like how are they going to respond to that? It is very it's a really interesting question. So thank you very much, Christian. It's been a wonderful talk.
Thank you very much.
We are looking forward to have you at the EBD in person once all the COVID-19 thing is.
I hope so.
Thank you so much.
Bye bye, everybody.